Hello Year 10, this is now the second revision video for the B1.3 unit, Digestion and the Digestive System in Humans. So in this video we're going to have a look at some past paper questions. What I'm going to do is to show you 2 or 3 past paper questions. I'd like you to try and write the answers down on a piece of paper in front of you, and then we will go through the answers. So the first question, as you can see, is on the screen. So this is showing you the liver and some parts of the digestive system. So I'd like you to have a go at answering those questions on the piece of paper that you have in front of you. If you need to pause the video as we're going through to give you time to answer the questions, please do. Okay, so question 2 again is concerning parts of the human digestive system. This one you need to identify 2 of the organs that perform particular functions and then label them on the diagram. Obviously you won't be able to do that on the screen, so either make just a little sketch of the picture on the screen and label it, or just have a think about where you would put the label for each of the diagrams. So the second part of question 2, a little bit more complicated because you're going to need to have a look at the results in the table and think about what those results are showing you. So read the information, have a look at the results in the table and try and answer those questions, please. Again, pause the video to give yourself time to think about what these answers would be. Question 3. Again, you've got some data there in a graph that you need to have a look at and think about what is happening. Okay, so if we now have a look at the answers for question 1. So you should have for organ X that this is the gallbladder. And Y is the stomach. For question B, fats are digested in the small intestine. Complete the sentence below. The liver secretes a substance called bile. This helps the enzyme named lipase to digest fats into fatty acids and glycerol. State the function of structure Z shown on the diagram above. Okay, so for this answer I'm going to show you the answer in the mark scheme. Okay, so what they're looking for you to say for that question, as you can see on the screen, is that the gallbladder carries bile into the small intestine, or bile travels or passes or flows through the gallbladder, through the bile duct. So the bile duct is carrying bile into the small intestine, or bile travels, passes, flows through, bile flows through. They're not going to accept that it releases bile, or just naming it as the bile duct, or saying that it connects the gallbladder to the intestine or carries bile to the pancreas. Okay, so what you're looking to write there is that the bile duct carries bile into the small intestine. Okay, so if we go back now to question 2, name the organ shown in the diagram above which secretes bile. Would be the liver. Label this organ on the diagram above. So this is the liver here. Name the organ shown in the diagram above which stores bile. Is the gallbladder. And again, label this organ on the diagram above. Would be here. The second part of this question then is a little bit more in depth. So you've got 3 test tubes here. Tube 1, they put just water and the olive oil. Tube 2, there was water, oil and bile. And then in tube 3, there was water, oil, bile and lipase. As you can see, the only place we're getting results is in tube 3. So the lipase is being broken down into its components. So that's why we're starting to see fatty acids become present. So what this experiment does is prove that lipase is needed to digest that oil into the fatty acids. So for part 1, bile plays a very important role in the digestion of lipids, but the result in tube 2 shows that it is not directly involved in the production of fatty acids. Describe the role played by bile in the digestion of lipids. So again, if we have a look at the mark scheme, what they're looking for here is for you to say that the bile is breaking down large droplets of the oil into smaller droplets. So that would be for the first mark, or just using the term emulsify. So bile is emulsifying the fat. They won't accept you to say that the bile is breaking down large molecules into small molecules, because that would be what the enzyme does. The bile is just breaking up large droplets of fat into smaller droplets. The second mark is for saying then that that gives it an increased surface area for the enzyme to work upon. So if we go back to question 2, the production of fatty acids in tube 3 did not increase after 20 minutes. Suggest a reason for this. So you've got 2 possible options here. You can either say that all the lipase, all the oil has been digested. Okay, so there's no oil left to digest, so obviously we wouldn't then see any more fatty acids. Or you can say that the enzymes are working as hard as they can, but you're better off really there saying that all the oil has been digested or broken down. Apart from fatty acids, name another project of lipid digestion which could have been tested for in this experiment. Would be the glycerol. Okay, so if we look now at question 3, why do we need to digest large food molecules, was your first question here. So if we look at the mark scheme for question 3, why do we need to digest the large molecules? they will accept any one of these answers. So to make them small enough to be absorbed, to make the food soluble, or so that they can pass through into the blood through the intestine wall. The second part of the question then, describe the effect of pH on the rate of digestion of the lipids. So looking at the graph, in acidic solution, we haven't got much digestion. In neutral solution, it has increased. And then in alkaline solution with bile, you can see we've got a greater rate of digestion. So as pH increases, as pH increases, you're going from acidic to alkaline solutions. As pH increases, the rate of digestion increases. And then finally, explain the effect of bile on the rate of digestion of lipids. So this is similar to the previous question. So the bile emulsifies the lipids, giving a larger surface area for lipase to work on. Okay, so we're going to do another couple of questions now and then look at the answers. So this is question 4. First thing you're asked to do, and this is bringing in knowledge now from previous units, is to state the meaning of the term diffusion. We've then got a diagram showing the similar experiment to one that we've done. So a piece of viscin tubing that's been set up to represent the way molecules pass through the wall of the small intestine into the bloodstream. So we've got starch and substance X in the water inside the viscin tubing. Then you've got water around the outside. Every 30 minutes for the next 2 hours, the water in the test tube was tested for the presence of both starch and glucose. So again, pause the video at any point if you need to try and read through the information and make sure you're understanding what it's saying. So the results are shown in the table below. Okay, write down your answers now for these questions and then we'll go through them. Okay, question 5, we've got a diagram of part of the digestive system. Right, this is still part of question 5. It's quite a long one, worth 12 marks. So read through the information carefully and try and come up with an answer. Okay, this is then question 6. Again, you're bringing in knowledge now from the first unit about enzymes to help you answer these questions. This is the last part now of question 6. Graph showing the effect of temperature on the rate of an enzyme control reaction. Okay, so if we go back to question 4, state the meaning of the term diffusion. What you'd need to say is that diffusion is the movement of either molecules or particles from an area of high concentration to an area of low concentration. So the molecules are therefore moving down a concentration gradient, but as long as you say they're moving from where they are high concentration to where they are low concentration, then that would be fine. So the second part of the question then, where they've looked at this experiment here, the first question asks why starch does not appear in the water in the test tube. So what you would need to be saying there is that the starch molecules are too big to pass through the pores in the Viskin tubing, or that the holes are too small for the molecules to pass through. So the starch molecules are too big to pass through the holes in the Viskin tubing into the water. So the next question asks you to name substance X and explain why glucose appears in the water in the test tube. So if we go back to the diagram, inside the Viskin tubing there was starch and substance X. Now for glucose to appear, the starch must have been broken down. Therefore substance X must be an enzyme that is able to do that. So what is happening here is that substance X is the enzyme amylase. It would be breaking down the starch into glucose. The glucose then is small enough to fit through the holes in the Viskin tubing. So if we look at the mark scheme, you can see that the first mark that's available for this question, so this answer is out of 4, you can see that the first is for saying that substance X must be either amylase or a carbohydrase enzyme. The second mark is for saying that the amylase would digest the starch. The third mark for saying that the starch would be broken down into glucose. And then the fourth mark for saying that the glucose then is small enough to pass through the pores or go through the holes in the Viskin tubing into the water. Finally then, for question C, suggest the temperature at which the reaction would work at its fastest rate by circling one answer below. We would need to circle 35 degrees. At 0 and 10, the enzymes would not have enough energy to effectively carry out the reaction, and at 100 degrees the enzymes would have denatured. So if we look now at question 5, the diagram shows the lining of part of the digestive system. Name the part of the digestive system that has this lining. This would be your small intestine. If the surface area of a single villus is 5 millimeters squared, calculate the total surface area of all the villi shown. Assume that all of them are complete and include a unit in your answer. So if you count how many villi are in the diagram, there are 14. So 14 times 5 would give you 70. So your total surface area would be 70 millimeters squared. They will also accept 0.7 centimeters squared. Question C, state 2 functions carried out in this part of the digestive system. So if we look at the mark scheme to see what they'll accept, here we've got absorption of digested food and digestion or breakdown of food molecules. You could name particular molecules, but they will not accept produce enzymes or just breakdown of food. So it's digestion of food molecules or digestion of carbohydrates, proteins, fats, and absorption of those digested foods. Part D then, state 1 feature of the digestive system which helps this part carry out each function named in C. So to help with the absorption of digested food, you could mention the rich blood supply or large surface area or thin walls of the villi. And for the digestion or breakdown of food, you need to mention that the small intestine is producing enzymes to digest. If we look at the next part of this question then, fresh pineapple is sometimes added to meat to make it tender. Fresh pineapple contains a protease enzyme. Alan investigated how this took place. He used the following. So fresh pineapple that had been heated to 100 degrees C and cooled, and a solid jelly made from protein called gelatin. 2 samples of solid jelly were made and the pineapple was pushed into them. After 24 hours, the results were shown below. So with the fresh pineapple, the solid jelly has become a liquid. With the pineapple that had been heated to 100 degrees C and cooled, the jelly has remained solid. Explain the results with the fresh pineapple. So with the fresh pineapple, the protein in the jelly, so the gelatin, has been broken down or digested into amino acids which can dissolve. So that's why the jelly has become liquid. So within the fresh pineapple, there must be enzymes which are able to break down the gelatin into amino acids, which has turned the jelly into a liquid. In the beaker where the pineapple has been heated to 100 degrees C, that will have denatured the protease enzymes in the pineapple. So the enzymes have been denatured, the active site has changed shape, so the digestion of the protein will not happen because the protein is not going to be able to fit into the active site of the enzyme. So we move on to question 6. Use some of the following words to complete the table about enzymes. So enzyme protease, substrate is protein. Your products here would be amino acids. Lipase substrate is lipids, and your products. So for this, there would be 1 mark for naming both of these products, fatty acids and glycerol. The diagram shows 4 enzymes, A to D, and 4 substrates, 1 to 4. So enzyme A would work on substrate 3. Enzyme B would work on substrate 2. Enzyme C would work on substrate 4, and enzyme D then obviously would work on substrate 1. So you would need to get all of those correct, as you can see in the mark scheme, to get the marks. So there wouldn't be any half marks. You need to get all 4 of them correct to have the mark for that question. So if we now have a look at the graph, so the graph shows the effective temperature on the rate of an enzyme control reaction between 10 degrees C and 50 degrees C. From the graph, describe the effective temperature on the rate of the reaction between 10 and 50. Now, it's really important when you get a graphing question that you read the instructions, because this one is asking you to describe. Lots of people will try and explain why this is happening, but it's not asking you to do that. A description, when you're asked to describe a graph, you literally just say what the graph shows you. So for this one, describe the effective temperature on the rate of reaction between 10 and 50 degrees. As the temperature increases, the rate increases for 1 mark until it reaches the optimum temperature, or 35 degrees C for the 2nd mark. It then falls for the 3rd mark. So you can see you haven't tried to explain why it's going up and then down. We're just saying what it's happening. So the rate of reaction is increasing until we get to 35 degrees and then it falls. Most enzymes are denatured by boiling. Use your answer to part B to explain why a denatured enzyme can no longer work. So again here you would need to say that the shape of the active site has changed, so the substrate can no longer fit into the active site. So the enzyme and the substrate are no longer able to join together. Okay, so we're now going to go on to question 7. So again, just think about where you would put these 2 labels. And then read through this information. So these are the results that she's got from the experiment. So you might need to rewind the video slightly to read what was on the previous slide and then come back to this one to help you answer the questions. Take your time. Pause the video. This is quite a complex question. Okay, question 8. We're going to have a practice at an extended answer question. So this question would be worth 6 marks. So describe the functions of bile and lipase in the breakdown of fats. So this is worth 6 marks. And you'll also be assessed in these types of questions on your quality of written communication. So you'll be looking at your spelling, your punctuation and your grammar. Okay, so we're just going to go through the answers to these 2 questions because they were longer and more complicated questions. So if we start with this one, on the diagram above, label the following. So this is the pancreas, and they will be specific in saying that your label line must touch the part to which it's referring. And then this would be the bile duct, where the bile flows through into your small intestine. Okay, so if we have a look now at this experiment, there's a lot of information for you to look at in this question. So in the exam you may want to highlight key points, make some little notes around the outside, and that's perfectly fine. So we've got 5 test tubes, with one with water, one with 0.5% amylase, one with 1% amylase, one with 1% amylase, and one with 2% amylase, but they're in different conditions. So most of them are at 37 degrees, apart from tube C, which was at 100, and all of them are at pH 7. So a pupil carried out the following procedure. She took a Petri dish containing starch agar, so a jelly in which starch has been dissolved. She cut 5 small holes in the jelly, so holes 1 to 5, and then she put 1 centimetre cubed of the solution from each of the tubes, into those holes. After 1 hour then, she flooded the Petri dish with iodine solution. So you should remember that iodine is testing for starch, and if starch is present, then the iodine will turn blue-black. So if we then look at the results, so at the start you can see you've got the 5 holes filled with the different solutions. They haven't told you which of the solutions is in the other holes. And then the second one shows the results after 1 hour. So the jelly, where there is starch present, has been stained blue-black by the iodine solution. So question 1, explain the appearance of the clear areas. In the clear areas, obviously there is no starch present, so the amylase has digested, or broken down, the starch. State the solution that was added to hole 2. Explain your answer. So hole 2 has the biggest clear area, so that is the one where the more starch has been broken down. So if we go back to the table, obviously we would expect that the one with the highest percentage of amylase would break down the most starch. So state the solution that was added to hole 2 would be solution E. So if we look at the mark scheme, tube E, or the 2% amylase, because it's got the greatest concentration of amylase, so it contained the most concentrated amylase. Not more amylase, because your volume would be the same, it's just got a higher concentration. So it contained the highest concentration or percentage of amylase. You were then asked to identify which hole contained the contents of tube C. Now, if we go back and have a look at what's in tube C, that was 1% amylase at 100 degrees and a pH of 7. So because the amylase is at 100 degrees, it will have denatured. So we would predict that it would be hole number 5 that contained the contents of tube C, because there is no digestion of starch occurring, because the enzyme had been heated to 100 degrees, which means it has been denatured. So the first mark is for identifying that it's hole number 5. The second for saying that there has been no digestion of starch, there's no clear area, because the enzyme has been heated to 100 degrees, which means it has been denatured. The final question here then says to state the purpose of tube A, the contents of which was put into hole 1. So again, if we go back, tube A had distilled water. So tube A acts as a control. So it shows us then that it is the enzyme that's digesting the starch, because nothing has happened when there is just water. So it acts as a control to show that it is the enzyme that digests the starch. Okay, so this question then, you were asked to do a 6-mark question to describe the functions of bile and lipase in the breakdown of fats. So this is the mark scheme for this question. So the sort of information they're looking for is for you to say that bile emulsifies fats from large droplets into smaller droplets, increasing its surface area. Bile then increases the pH or neutralizes the acid stomach contents, making it more alkaline, less acidic, creates an optimum pH in the small intestine for the lipase enzyme, which digests or breaks down fat into fatty acids and glycerol. So if you've got all of that information down, then but as well as having all of that information included in your answer for 6 marks, you would also need to look at the information below. So for 5 to 6 marks, you need at least 7 of those points. So at least 7 of those bullet points above. But you also need a sustained line of reasoning, which is coherent, relevant, substantiated, and logically structured. So basically what you're writing makes sense and is in a logical order. You've used appropriate scientific terms, so you've used enzymes, surface area, fatty acids, all of those, and accurate spelling, punctuation and grammar. To get 3 to 4 marks, you need to have at least 4 of those bullet points, and your writing would be partially coherent, so it mostly makes sense and is largely relevant, supported by some evidence and with some structure. You've used mainly appropriate scientific terms and some accurate spelling, punctuation and grammar. For 1 to 2 marks, you've got at least 1 point from the indicative content, so from the bullet points above, but the information that's been written is quite basic and maybe there's some information in there that's not relevant, supported by limited evidence and with very little structure. So there'd be not many scientific terms and inaccuracies in spelling, punctuation and grammar. So look at what you've written down, think about which of those categories you would fall into and give yourself a mark out of 6. Okay, we're going to move on now to question 9, which you can see there on the screen. So again, write the answers down, pause the video if you need to, and then we'll have a look at the correct answers. Okay, this is still part of question 9. And then the last question we're going to have a look at is question 10. Okay, so if we have a look at the answer for question 9, during digestion in the human body, large food molecules are broken down. Draw lines joining the large food molecules to the smaller molecules into which they are broken down. So protein is broken down into amino acids, starch is broken down into glucose, and fats are broken down into fatty acids and glycerol. Why is it necessary for these large food molecules to be broken down? So if we look at the mark scheme, they'll accept for that one for absorption, so they can pass through the cell, for getting into the blood, or for use by cells, or to pass through cell membrane. But your best answer there really is so that they can be absorbed through the wall of the small intestine. State the function of carbohydrate foods in the human body. So if we look at the mark scheme again, for, to release, to get, a source of energy. Okay, so they provide energy for the body. They will not accept, though, to produce energy or make energy, because as you should know, energy cannot be created or destroyed. It can only be changed from one form to another. So what we're doing with the carbohydrate is to release the energy that is inside. From the diagram opposite, give the letters which show the pancreas. So the pancreas you should have is F, and the large intestine is G. Give the two letters which show where protein is digested. So protein is digested in the stomach, which is E, and the small intestine, which is C. Last bit of this question then, the food substance glucose, the test solution would be Benedict's, and the food substance which would be tested for with Biuret solution is protein. Our last question then. The following statement refers to a process that occurs in the digestive system. The muscles in front of the food relax while the muscles behind the food contract. Name the process being described. You should know that this is peristalsis. Question B, you're looking again at a graph. So at different pHs, mass of food undigested. State what happens to the mass of undigested food from pH 3 to pH 6. So again, it's asking you to just say what's happening rather than explaining why it's happening. So as it's worth 2 marks, that would suggest that you need to make 2 points. So the first mark is for saying that it increases until pH 5. And then it levels off. And finally, state the optimum pH of this enzyme. So the optimum pH would be pH 3 because that's where we're getting the most digestion. Name the organ in the human body where this enzyme is found and name the class of food it acts upon. So this would be found in the stomach because that is where you would have the acidic conditions. And then the food substance which it would work on would be protein. Okay, so that's the end of our video now, looking at some past paper questions.